If health is wealth, then AI is helping us get richer. From improving clinical care to empowering personal health monitoring, AI-driven solutions are taking part in changing the way we get and stay healthy. AI has found itself into different aspects of health and wellness. From molecular diagnostics and genetics to radiology and orthopedics and even mental wellness, AI is revolutionizing healthcare services in ways we can never imagine. AI's role in the Philippine healthcare system continues to grow as medical services are pushed to be better and more accessible to the Filipino public.
I joined the Philippine Genome Center last year, coming from the States for practicing in the States for 32 years. And I think it was calling and giving back that made me come back. I've been with the Philippine Genome Center as a scientific advisory board for health since its inception. I think this is one kind of example that there's hope, because 15 years ago, if the EMR was on a bill but never really realized, the Philippine Genome Center also started in 2009. So Philippine Genome Center pledges a deeper understanding and judicious application of advanced knowledge and emerging technologies in genomics and bioinformatics for the development of either diagnostics, DNA forensics, improved crop varieties, preventative products, as well as therapeutics. And this is like an example of how cross-sectional we're going, because it's the same technology that we apply for both humans, animals, and plants. Okay, so I mentioned that the Philippine Genome Center started as a concept in 2011, I believe, and then at least for now, we're operational, and the pandemic was a silver lining because it actually underscores the role that the Philippine Genome Center had. Just to introduce how the Philippine Genome Center activities are, we have technical services as well as training and human resource development and research. So we assist all of other research groups in design as well as implementation of the research, but at the same time, we have technical services, and I think this is one of the one part of the concept paper that I've submitted to the President, wherein we're trying to evolve in such a way that we will be relevant to laboratory diagnostics. And not only that, but I strengthen our capabilities in bioinformatics as well as laboratory diagnostics so that we can reach out to a lot of people or the whole nation. I wanted to highlight how the core facility for bioinformatics is. We received some grant, or at least the BOR had actually given us or approved money for funding for the core facility, and we're trying to transform it in such a way that we could actually support both research and laboratory diagnostics. So giving the Philomics as a name for the core facility for bioinformatics, they've done a lot. And so we're trying to, and kind of like my vision is to keep all those people intact and retain them, because this is the hope of however Philippine Genome Center will evolve. We will then serve more and create more tests and offer it to the public. So I wouldn't go around the diagram, but practically we have objectives for research and extension, and at the same time we have actually ongoing studies on other sequencing for different organisms, including rice and other plants as well as humans. Very proud of this. When Dr. or VP Leo had asked us what has been the activities of the Philippine Genome Center for the past year, we were kind of like pleasantly surprised. We've done a lot, right? In a way that we've sequenced a lot of these organisms on the diagram, but at the same time, we're still just starting whole genome sequencing. And then, of course, the Philippine Genome Center was a very active, and it was the epicenter for biosurveillance for the COVID pandemic. It actually had identified or detected SARS-CoV-2 global variants of concern. It had submitted all this data into the consortium GISAID, and actually were contributing data globally. So we also had tracked SARS-CoV-2 transmission in the country, and you will be surprised. I'm not going to give you details, but actually this group had produced a lot of materials for COVID, and also have maintained actually the data in the center, and had fed the DOH Epidemiology Bureau, and identified local signatures of evolution of emergence in terms of new variants for SARS-CoV-2. So I would be remiss if I don't give you a little bit of background on genetics and genomics before I give you what are the challenges and what are the solutions that can happen, right? So forgive me if this is more science than anything else, but this is how it is, right? PCR was not even known to the lay person until COVID hit. And so therefore, if we're going to do Genomics Philippines, or we do whole genome sequencing for humans, you have to know the basics for genetics and genomics. Okay, sorry, but this is a very common, familiar diagram in genetics and medicine. It's a book that gives you all the basis for genetics and medicine, but it's actually a very simple one. Every cell in our body, you and me, every cell, we have a nucleus that actually contains the blueprint or the genetic material that tells the cell to function how it should be, right? So then how does it connect? The genes, the protein is the product of the genes, and then the protein has this assembly, cell networks, or even signaling each other. And therefore, we are whole, we're functioning healthy, and therefore there's a genotype. Genetic information gives you the phenotype. So color of your hair, the color of your eyes, and everything else. Okay, so the genes and gene structure. There's like a very organized way in which the DNA is actually put together, and one can look at chromosomes. So the chromosome, or the karyotype, is like one simple way of determining how many chromosomes does this individual have? Is there a rearrangement? Is there a deletion or something else? But at the same time, it actually gives you a raw kind of picture on how that cell is working, right? In cancer, you would have some chromosomes that are missing or some deleted, and that's like one kind of like scanning passing through in terms of what happened to the chromosome. And then going down to the level of the gene, one can have, any gene should have this structure, right? So you have a regulatory part, and then the express, which is actually, that's the one that's coding your protein or amino acids for your protein, and then the introns, which are regulatory, meaning that we cannot be all exons, or a gene cannot be all exons, because it would be subjected to a lot of pressure, and any mutation or change in the language will cause problems, okay? So that in itself, the genetic information that's on the gene determines the protein, amino acid, and then the tertiary and the secondary, tertiary, and quaternary interaction of those proteins. So function is determined by the DNA. Okay, so I also would be remiss if I didn't tell you this extent, right? Because you will hear transcriptomics, metagenomics, epigenomics, all of those things. But actually, it's like, it's a process, right? Sabi ni President Gigil, intimidating maybe, but not really, right? Because eventually you should have a kind of like, even clinicians have this different knowledge base, but at the end of the day, you will have to have a basic understanding, because cancer is one good example. It's a genomic disease, and practically one can understand why there are problems, and there are treatment bases for each of those management. So I mentioned the cell, right? So there's extracellular proteins, there are cell surface and nucleus, but I'm not going to give you a lecture in molecular biology or anything, but just to say that there are those that are actually controlling each function within the cell, and that actually can go awry and cause problems. I'm not giving you all the biomarkers, but actually cancer is a very complex disorder, right? So any tissue can go into cancer or transform into a cancer, and practically there are biomarkers. And that's where genetics comes in, because it could be an acquired one caused by environment, or it's something that's genetic or constitutional to the individual. So yes, there are hallmarks of changes, and it could be an early detection hallmark for as a biomarker for cancer, or it could be far along, or it's something that one can use for treatment. And I will tell you, breast cancer especially, right? It's like one of those prototype of breast cancer is not just breast cancer that is somatic, or meaning that you acquired. It's actually background of genetics that's actually, you have it in you, but it could be environmental effect on the cancer that you have. So liquid biopsy, I know this is like the hype, right? So if you get a sample of blood, you'll be able to detect what's wrong in terms of some of the drivers of cancer. But this is not early detection. It's still far from you being utilized as early detection, because it has limitations. So what do you get out of the blood that you get from a patient? You will have total RNA, total DNA, and therefore you can look at what is constitutional and what is actually cellular, meaning that what did the cancer cells really produce, right? So transcriptomics then comes in, because this is RNA, and you are looking at your the cellular products, or the dying cancer cells that are emitting or releasing all these transcriptomes in the blood. This is being used now for monitoring. So if you have a cancer, specific cancer, colon or breast, they could look at your blood and see where the driver mutations are, the variants that causing your cancer. And therefore, if they target that variant, or there are some specific drugs that actually would target breast cancer cells, then you would know whether there are residual or monitor the cancer cells. Okay, again, this is like, we know that we can detect them because platforms, the technology had already evolved, right? In fact, I was a clinical geneticist in late 90s, wherein we only had one or two tools or platforms that can diagnose a patient. So karyotyping or chromosomes, and then single gene testing, right? Right now, there's a lot of platform, next generation, transcriptomics, epigenomics, all of those other things that actually are available. I've seen it rise in terms of advances, and practically from research to clinic, and practically also looking at one patient that doesn't have a diagnosis into a patient with a diagnosis because of all the advances in genetics. And then I'm a pathologist. Maybe some of the people in the audience are pathologists, but actually cancer, there's a lot of technology that has actually been advanced because of cancer, right? So pathology look at the tissue itself, like what's being shown on the left upper. And then we look at the tissue under the microscope, and you look at it in such a way that, oh, there's something wrong in this tissue. It doesn't look normal. And then you use some other ways to really confirm if there's something going on in that tissue. So those other stained slides there are saying that, oh, yes, this is expression of a protein that should not be there or expressed too high, and therefore this is cancer, and therefore the grade of the cancer is higher. So those kind of tools are in pathology, and they're being used right now as part of an integrated report where you have the genomics, genetics testing, plus this histologic morphology. We also have cytogenetics. I remember the chromosome that I've shown you earlier. They are like coiled DNA, and so they aggregate, and practically you can use some ways to detect or hybridize a DNA of the same or complementary strand, and you would know exactly what is missing and what is not there in terms of cancer. So this is called cytogenetics fish or in situ hybridization, but it gives you a visual. This is like the drone trying to look for changes, and actually optical density, optical changes can be utilized for diagnostic purposes too. And of course, DNA sequencing history, I started with the slab gel. Doing research on slab gel is way really harder than running a sequencing in a 96-well or capillary sequencing or even next-gen sequencing. So practically, it gives you more information at the same time, less tedious and more efficient. So what actually influenced genetics and genomics testing? It was too expensive to sequence, right? Imagine the slab gel. We can't even sequence probably 2 genes in one slab gel. And then for capillary sequencing, you can sequence a couple of genes. And then came the next generation sequencing, wherein you can actually sequence whole exome sequence or whole genome right now. So practically the cost per base pair sequence is actually pushed down because of technology, and therefore now it became more readily available in the clinic. With all the next generation sequencing advances, we now know that there's probably 22,000 genes in each of those cells that we have, but we only know of the function of a third or a one-fourth of, one-third of those genes. So there's a lot more to discover because we don't know a lot of the functions of those genes. So we continue to use them in the clinics anyway because we have patients that would need them, and then identify those diagnoses if ever they have implications for the family. So the utilization of genetic and genomics testing, it's practically driven by utilization, technology, and science. Science, I showed you the karyotype, which actually would only diagnose numbers of chromosomes, and trisomy 21 or Down syndrome is a technology way back in 1959. And now where are we? We can now do transcriptomics and liquid biopsy. So in terms of utilization, evidence and clinical utility is very important, and at the same time, accessibility and affordability. This is not also, it's something that even the advanced world or the Western world had experienced, right? So I practiced in the US for 32 years. We didn't have all of this technology too. There's no adaptation because it's expensive, right? But right now, here's how the evolution of clinical practice is. So way back when we did testing, and all those genetic tests are being ordered by clinical geneticists because they're the only ones who understand how to order this test and how to interpret the results. But right now, I'll skip the middle part because right now, next generation sequencing had already advanced diagnostics or even recreational genetics, and now a patient can request for a next generation sequencing for whole genome sequencing. So what will you do if the patient comes to you and say, "Doc, here's the results of my whole genome sequencing"? How would you answer all those information, right? So that becomes, yes, consumer direct testing. However, there are people who are very curious about their genomic makeup, and they would really go to the extent of paying how much, 100 or hundreds of pesos, hundreds of thousands of pesos to get their whole genome sequencing. So clinicians in the group and even any other, we have to be very careful in terms of really utilizing whole genome sequencing, but at the same time, we cannot be left behind. Our neighboring countries in Asia are already far ahead of us. Vietnam had become a bioinformatics hub, and even the States, they know that Vietnam is like way ahead of some of the other centers, and we cannot be lagging behind. So for genetics and genomics testing, I think one needs to understand that it has to be created by professionals and practically utilizing cutting-edge technology, but it has to have a meaningful application. You cannot just sequence just for sequencing's sake, but actually there should be a purpose and there should be a follow-up in terms, or even from the very beginning, you have to consent your patient or consent the individual, because there would be some findings after you analyze analyze the sequencing, that would be very hard to explain, and I will get down to that. And therefore, not even geneticists can explain it properly, and therefore, those are gaps within the testing. Then it has to be related to the test or clinical practice. Cancer genomics had really explode, I think, because there are drugs that are really tailored fit for certain variants of a cancer gene, right? So then it becomes like, if you have this mutation in your gene and you have breast cancer, you can be treated with this kind of drug, and therefore there's remission and there's hope for the patient. Okay, so genomic centers of excellences have really evolved, and then practically experts, right? There are MDs, MD PhDs, but the point here is like, it's an evolution. We cannot have everything, but at least in the Philippines, if we start workforce development, and we actually retain our talented MD PhDs, and MDs here in the Philippines, we can come up to a point where we could be competitive not only locally or regionally, but also globally. Okay, so a little bit of next generation sequencing and where AI will come in. So I said we have, how many? 3.6 trillion base pairs, right? So practically that's like something that has 3.4 gigabytes when we sequence it. But at the same time, I'm showing you how next generation library preparation as well as detection. And it's also like showing you that the colors are very important. Optical imaging is very important. And it's the same principle that a lot of our other AI equipment or gadgets within in agriculture and all those satellites are looking at, it's similar, right? But it's just going into the cell and making it more really truly hyper high resolution, so we get the sequencing or the base pair makeup. Okay, so in pathology, we have pre-analytic, analytic, and post-analytic as a phase in terms of a workflow when you have a sample. Remember, each one of you probably had done blood draw, and during blood draw, this is like the process, right? But in genetics, we practically get a clinical sample, and we need to get a clinical insight. And so that, whatever that analytic part is, whether it be a tissue, a histologic examination, a blood examination, or genomics or genetics sequencing, someone had to do it and come up with that workflow. So I will probably skip this for time, but bottom line, during the processing of that sample from a patient, be it a tissue or blood, somebody needs to understand that there are information that needs to be processed. So data management is very important. You need a laboratory information management system that tracks all your samples. You need information data management that would really capture all the results, and then go in through whatever machine learning or AI that needs to happen in terms of bioinformatics processing, and get to a results. What do you call a results? It's actually a variant call. And if that variant call needs to be curated and annotated, it needs a lot of information. Imagine if you have, an individual would have at least probably 1,000 variant calls. Then you have to make sure, are all these variants relevant? And then, of course, you need to know your medical or your clinical information, because the clinical information gives meaning to whatever variant you found, right? So that is actually AI in a way, but it's not perfect yet, and it does need a lot of data mining. Some of the informations that are actually out there are databases that are open source, and as well as curated by very highly STEAM groups in the US and in Europe. They practically get to a point where Filipinos are not represented in the human genome sequence assembly. I mean, if we are ever represented, it would be very few of us in that human genome sequence assembly. So I'll give you an example use case. My sister got breast cancer at 50 years of age. I said, genetic, right? Because it's 50 years old, you suspect genetic. be it a background or something that she acquired. I went through the motion of having her blood sequence and as well as her tissue sequence in the States. I spent, say, $200,000 at 200,000 pesos in equivalent, right? But giving it back to her here in the Philippines so that the oncologists can take care of her is something else. But at the same time, she had a variant of unknown significance in BRCA2 or breast cancer gene 2. What is the explanation? No one in the States would give me an explanation or variant interpretation. Then I asked geneticists in the Philippines, I said, do you have this patient who have undergone breast cancer testing? Does this variant really significant in Filipinos? No one knows, okay? So then it becomes now a search of what is this? What is this? And even our family, practically, it's like, it's a psychological kind of burden because I'm a geneticist. I know what it means in a way, but I don't really, and I can't explain to my family and how we would interpret that variant of unknown significance. So I use this as a use case wherein we need our own Philippine genome represented, or if even better, we create our own human genome Filipino assembly so that we can interpret those results precisely and accurately and help individuals like my sister, or like our family. So here's like a workflow. It's very tedious, but at the end of the day, you have to have a result that is meaningful. And this is like a cartoon that gives you all the, in summary, what are those variants, right? It's a very dynamic process, and it requires interpretation. You have to go back to the literature, iterate, look for that variant again and again, because it changes with more information and more patients being sequenced. I will give you like a quick use case here. I was with the largest reference laboratory in the US, and this is actually published, so I can divulge, right? There was one patient who had epilepsy and had a mutation in one epilepsy gene 10 years ago that was interpreted as benign, meaning it is not causing disease, okay? 10 years after that, it was changed into pathogenic variant, meaning that it's now telling the clinician that that variant, which was interpreted 10 years ago, was actually the cause of the disease for that patient, okay? So now the family sued the clinician, right? So now it becomes like, yes, we have to realize that there's a dynamic way of interpreting results and there are limitations, and therefore, right now, every molecular diagnostic lab will put it under the interpretation that this interpretation may change with time, okay? So that involves a lot of data mining, a regular data mining, and I was like, just kind of like, oh my God, somebody needs to help Philippine Genome Center because we don't want to get to that problem. Okay, so just to show you that there are guidelines, and this is a very practically very systematic way of variant annotation. And in cancer, it's more complicated. There are guidelines that actually give you interpretations for the gene that you're looking at, what is the significance, what is the evidence, and what is the treatment. So cancer is a gold kind of like, it requires a lot of information, and it also heightens the fact that there's a need for clinical information and laboratory information to be actually integrated. Just to show you that there are databases out there, here are some of it, and I don't want to belabor it, but again, are they interpreting Filipino genomes or Filipino genetic testing? Here's some of the types and sizes, and this is something that comes out of next generation sequencing. And for those people who are looking at big data, how big is the human genome, here's how it is. So if it's a FASTQ file, you have that 50 gigabyte for an exome, but 18 gigabytes for 4,800 genes, and goes on and goes on. So practically, data storage should be a problem. Once we initiate whole genome sequencing, data storage would be something that we need to address. So I also had said that omics is coming. So it's not just the DNA sequence that we're going to do. We can do proteomics and correlate proteomics with the genomics, and all those transcriptomics also can be part of this omics. In that is going to cause us problems, but brilliant people within Philippine Genome Center had thought of, we need to have an interoperable health record as well as interpretation of all this genomic data. And so we proposed to the PCHRD that we should have not only a laboratory information management system, but system that actually can talk to an EMR in a different hospital and be able to mine whatever information we need to use for data or results interpretation. So this is actually going to be one of the things that needs to be funded. I think it is funded for 1 year, I think. And then practically, because we need to bring in the test for us to do it, PGC, we're actually poised to partner with an outside third-party laboratory that would transfer technology to the Philippine Genome Center. So considerations and challenges. I don't think I will have to enumerate everything, but bottom line, there is some infrastructure. It's not the perfect set for us to use for a laboratory service. However, I think among the different laboratories in the country, Philippine Genome Center is still the one that could lead all the advances in genetics and genomics technology. And yes, there would be cost for each of those development that we need to have, but we are being creative in such a way that we will get those infrastructure ready for lab services so that other parts of the country can be, or all of Philippines can be served, because we have PGH Mindanao, PGC Mindanao, PGC Visayas, and PGC Los Baños. So that, I think, opportunities and solutions, we need to start somewhere. We need to educate co-clinicians and co-MDs here, but also enticing people to participate in the whole genome sequencing of the whole Philippines. We call it Genomics PH, so that we get to be represented in the database and we can interpret results better. So I wouldn't also read all of those post-analytic solutions there, but equitable, accessible, affordable tests, we hope we can give it or provide this to the country in the next 2 to 3 years. Workforce development and talent retention is something that from day one when I joined PGC, was a concern of mine, but I think that there should be some ways to really attain full development as well as talent retention, and then streamline procurement processes, because we're going into a laboratory service lab that needs to be functioning and really sustaining all those activities in a turnaround time that's reasonable. I know I have used much time, but I will go back. I will actually wanted to give you what the President had mentioned earlier. We're embarking on Genomics Philippines initiative. This would be a 5,000 whole genome sequence for 5 years. This would help us in the variant database that we're going to build. It would help us create those infrastructure that would really sustain the laboratory diagnostics part of Philippine Genome Center. And then we will have our own Filipino database and we can interpret our results. Okay, so then the last portion of this slide, we have to establish the Clinical Genetics Genomics Laboratory for Diagnostic Services. We're starting with cancer, partnering with a third party outside of the country, and also putting the infrastructure of the laboratory information management system, database, even biorepositories. So we're trying to complete the whole kind of like facilities so that we could really serve the country the most. Okay, I think I will end up with bioinformatics and AI, and practically I sort of alluded to how we do those variant curation, but reiterate what Dr. Dance had mentioned, right? So there are opportunities for applications for AI or augmented intelligence, and therefore we have to start somewhere, and I think that we hope the Philippine Genome Center can maximize and utilize whatever infrastructure is available and then build brick by brick what we would need to have in the future and be really able to serve the whole country. I wanted to, never mind that, takeaways. Genetic tests are not created equal. So practically, the clinician who's going to ask for genetic testing needs to understand that there are limitations to each of those testing. It has to be something that is guarded and discernful, and then practically the clinician is the patient advocate. It's very expensive. Cancer, I gave you the figures for my sister, but practically we would reduce that cost when we open our genomic laboratory services because it's economies of scale. Right? So if we're serving the whole country, if there is more than 1,000 cases that we would serve for 1 month, and we're able to really drive the cost of sequencing down, then we're happy with it. Again, the right patient, the right test at the right time, very important. And then, of course, our baby steps in the transformation of the laboratory services towards an accessible, affordable, and equitable genetics genomics testing for Filipinos. And then the last one, I think it's not just my crusade, it's our crusade, and we would need collaborative partners and stakeholders so that we could advance the test development that are relevant to practice of precision medicine in the country and push on omics in the Philippines. Thank you.